for joining us today for SLAS's Technology Idea Exchange, a future for drug discovery. We're glad to have you all here, and we look forward to this conversation. To kick us off, our scientific director, Emily, is going to do some introductions, and then our presenters will go through their presentation and allow time for discussion and Q&A at the end of the presentation. Emily?
Thanks, Gina. Hi, everyone, and welcome to today's Technology Idea Exchange. We're super excited today to bring you a Technology Idea Exchange featuring 3 companies from the inaugural SLAS NexusXp People's Choice Award winning collaboration, with the test portion of the collaboration presenting on the future of drug discovery, DeepCure's AI and robotics driven pipeline. As Gina mentioned, my name is Emily Yamasaki. I'm the Interim Scientific Director here at SLAS, and I would like to also briefly introduce our panelists for today. So we have Thrasyvoulos Karydis from DeepCure, Andrew Leightner from Waters, and Dustin Whitson from Biosero. And we'll also have Hayley Dyer from Biosero moderating our discussion today. So to get us started, I will hand it off to Thras to kick things off with a short presentation from DeepCure.
Excellent. Thank you so much, Emily and Gina. And thanks everyone for the organization of this webinar. Super excited to dive right into the topic of today. And before I do that, let me share my slides. Great. Are the slides going through?
They're showing the presenter mode right now. I don't know if you could switch it.
Oh, yeah, I can.
There you go. Perfect.
Perfect. Great. So, hi everyone. I'm Thras from DeepCure. Today's topic is going to be all about automation and AI and how they go hand in hand in our attempts to actually go to build the future of drug discovery. I wanted to start briefly by introducing DeepCure because we are, a young startup started out of MIT around 2019 in Boston. We're a therapeutics company. We're working primarily on inflammation and autoimmune therapeutics. Inflammation is meant to be the most significant cause of death, as we have an ever-aging population. And our approach from the start has been to work on problems that are quite challenging for what you would call the traditional small molecule discovery. And our current pipeline actually is at a stage where we have proven multiple times that we can generate small molecule therapeutics for targets that were considered intractable. We have our BR4BD2 program with an extreme selectivity profile that enabled it to go all the way to the IND enabling stage, where more than 137 clinical trials have failed in the past. We have our STAT6 program, which some of you may know is one of the highest grossing drugs at this point, the antibody dupilumab. We have an oral alternative that's as efficacious and safe. That's now on the way to the IND. And of course, TNF-alpha highlights our ability to be able to target things like protein-protein interactions. And all of these problems, straight from the first few years of DeepCure, highlighted the need to be able to make a lot of molecules. And not only make a lot of molecules like a high throughput screen, but make a lot of the molecules that our AI design tools dictated for us that they would work for the targets. So another big result we had in the past couple of years was that we built an automation platform that we now call Inspired Chemistry that was able to rapidly make complex and diverse compounds that were designed by AI. And what I mean, the prototypical case there was being able to make one of the active ingredients in Paxlovid, a very complex molecule, 6 stereocenters, and multiple synthetic steps. And the way that we made it, the process enabled not only synthesizing the exact compound, but quite seamlessly synthesizing 56 diverse analogs, introducing diversity at every synthetic step within 30 days. Before I go into the how, I wanted to add a little bit of high-level motivation on why we need automation and why we need not only just any automation, but automated chemistry synthesis that can give us diverse molecules. Here I have a statistic that actually is computed by the graph of how many approved drugs for new targets per year come out in the global market. And this number is 15 to 20 drugs. Now, if you take that and take also the number of targets, proteins with potential therapeutic effect that we know of, which is more than 8,000, you get this number. It would take more than 400 years to even start exploring therapeutic opportunities for all the targets that we know may have a therapeutic effect. So when I'm thinking about how DeepCure, how AI can accelerate drug discovery, I'm not necessarily thinking about how to go from 1 year or for 3 years to 1 year, but what I'm thinking about is how can we get this 10x improvement on this number? How can we go from 400 years to 40 years so that everyone here in this call essentially may have a chance to have a cure for their disease in their lifetime? And to do that, we need to overcome one of the biggest bottlenecks, in our opinion, in what we would call traditional drug discovery. And this is the bottleneck of reductive thinking, or of a reductive process. Every single step across the discovery cycles, the design, make, test, analyze that you're familiar with, is built in a way that reduces the chemical space as much as possible to go to the next step. And the fact that the process is manual, is still manual to its biggest part, is at the core of that reductive thinking. You're going from being able to design virtually any molecule in silico and evaluate it, to very critically thinking, okay, how many of these molecules can I actually make? How much money and time am I willing to spend to be able to get back my data? Even on the testing, you use some assay that's off the shelf, or you have to spend maybe months on building a new assay, and this assay that's off the shelf does not allow you to explore this new, let's say, binding pocket on a protein. And then, of course, the software that we use to analyze has been also so fine-tuned and offers filtering operations that are extremely quickly reducing the diversity of solutions towards the one potential preconceived idea that you have on how to bind and target. So what we're thinking at DeepCure is, how can we actually change this process and make it from reductive into an expansive thinking process? Essentially, how do we start with what we know, whether this is some of the results of the AI tools that we use, or prior literature, or the idea of your structural biologist on how to go about a target, synthesize some molecules, and then once you get the results, start an expansive process where you now take what you know, take the things that worked or not, and you go back to the chemical space and try to find not only similar molecules, but different looking molecules, different types of ideas that can satisfy the data that you, or the hypothesis that you can generate with the data that you got. And then in the end, we believe, and actually we have seen internally, that this diversity is what's going to get you to your outcome. I'm sure many people on this call can appreciate the fact that drug discovery is not a linear path. You don't start with what you want to do and you end up with a drug. You usually figure out things about your protein or your program along the way. And then it's that serendipity, if you would say, that curiosity that's driven by the data that gets you to actually make the molecule that ends up in the clinic. How can we do that, though? How can we actually make a lot of data that for very different molecules in a cost and time frame that's meaningful for a drug discovery program? And of course, automation can actually answer a lot of these questions. AI needs a lot more data that are consistent, that are significantly cheaper, and you can get them in a fast time frame. And automation, if done properly, can actually give you all of that. But there's a caveat there. It's not all automation or just applying high throughput technologies that can get you there. There is already a significant amount of chemistry being done with automation, but that chemistry typically is in the form of library chemistry or parallel synthesis that has very little diversity, or lacks the diversity and complexity that our models are thinking at to be able to solve the discovery problem. Then there is, of course, manual synthesis, which is at the other end. You could make theoretically whatever molecule you want. You can spend a PhD on it, but it would take a ton of time and at least a few $1,000s per molecule. So what we're looking at automation to be able to do is to get enough chemical diversity and complexity that's relevant for the AI drug discovery era at a cost and time that's matching some of the extremely fast, a week and $100 data points that you can get for library chemistry or single-step chemistry. And to end and kind of trigger this conversation around these ideas, I would like to present a few of the things that we did at DeepCure to be able to get to where we are today with our pipeline. At the AI front, we built a ton of AI tools actually in the past few years. I highlighted here two that are at the core of this idea of generating diversity. Our first tool, Pocket Expander, is a tool that essentially takes a protein and allows us to forget any existing data for this protein. Because typically, existing data are there because they have probably led to a failure at some point. And that's why drug discovery is hard. Ignoring the existing data, we use Pocket Expander to figure out completely different ways that we can interact with a protein. It may be still at the same active site or protein-protein interaction site. It may be on an allosteric site. What we get out of it is, if you would say, recipes for types of molecules that can work for the problem we have at hand. And that's coupled, of course, with Molzen, which is kind of our next stage database of molecules. And this database of molecules is actually actively generated based on a hypothesis using generative machine learning or reinforcement-based machine learning. The way we build Molzen actually has automation automation in the back of our head. We built it so that it learns organic chemistry rules. It has a set of reactions that it can use and a set of building blocks that it can use. And the way it's building molecules generatively within the hypothesis makes it such that the final molecule has an exact recipe, an exact synthetic route that we can move on to the next step, which is our automated platform, to be able to make it and transform the virtual designs into actually testable hypotheses in the lab. So we've built a few different iterations by now of a high throughput automated synthesis, multi-step synthesis. This is I think, the prototypical implementation, and it contains, I would highlight here, 3 big pieces. A liquid handler. A liquid handler is absolutely necessary because it's the highest throughput machine that you have in the industry. And a lot of the work we've been thinking about is how do you fit chemistry into a liquid handler. It contains a purification system because being able to do multi-step is absolutely necessary to get the diversity that you want. And of course, it contains a scheduler/orchestrator, here in this case from Biosero, which allows us to essentially not have to go at 5 a.m. in the lab and take a plate from a place to another, or be able to, do all of the decision-making in a way that can be learned by AI. And to end this short presentation, which I hope starts triggering a lot of questions for the discussion, I want to say that apart from the hardware integration, which is critical for a project like this, the software integration is at least the same amount of important when you're building this. And we've built a piece of software we call Galactus as part of our bigger Inspired Chemistry platform. Galactus essentially is deeply integrated with both experiment orchestration and scheduling of the instruments, and with software that can give us analytic data, like Waters mass links or fraction links. And the importance of that is dual. One of them is what I was saying before, be able to automate as much as the human interaction so that we can operate 24/7 and increase the cost and time margins on delivering the compounds. The second piece, which I'm pretty excited about, is that we built a structured database for every single piece of chemistry that we've run in this machine. And then we can use that to actually train machine learning models that are able to take the decisions for what to do next in your assay. They are able to take, for example, the purification data from a reaction well and be able to take the decision, do I move forward to the second step of synthesis, or do I discard this so that I can save time and money? I'll end here and pass it on to the fellow speakers of this panel, and I'm glad to answer any questions about our system.
Ross, thank you so much for just setting the stage for the discussion with that presentation. The goal of the rest of the webinar is to just have an open and discussion style conversation. So we wanted to make sure to introduce this project to you and then be able to just go into something a little less formal. So I'm going to facilitate the questions for our panelists, and I'm also happy to take any questions that you have on the audience and then ask those of our team here. So if you have a question, we really want to hear it. There is a Q&A widget in the menu that you can use to send those questions over. So I'll be watching that widget as we go. We also received a few questions during registration, so I've got those in the mix as well. And to get us rolling, I just want to ask Dustin and Andrew to introduce themselves a bit so you have a sense of all the people on the panel. And both Dustin and Andrew supported Thras and the DeepCure team on their projects. So we're going to be able to talk about the collaboration between the companies as well. So from there, Dustin, who is from Biosero, he's an applications consulting scientist. Dustin, could I ask you just to say hello and just say a little bit about what Biosero does?
Yeah, sure. Thanks, Hayley. Like she said, I'm Dustin Whitson. I'm an application scientist here at Biosero. Our company develops science-centric software and lab automation solutions that really allow researchers like DeepCure to orchestrate their discoveries at every stage of the process. So we provide solutions for automation platforms ranging from very small systems with maybe one device or robotic arm, all the way up to biofactory level implementations with many work cells working together to achieve a workflow. Our expert team is happy to design, build, and deliver these systems around the world.
Awesome. Thank you, Dustin. And Andy, last but not least, could you introduce yourself and talk a little bit about what Waters does?
Absolutely, Hayley. Thank you. So my name is Andy Leightner. I am a Senior Principal Market Development Manager with the Americas team, focusing on pharmaceuticals and biopharmaceuticals. For those who might not be familiar with Waters, Waters provides the analytical tools, software, and consumables used by chemists to help discover, develop, QC, and support the continuous purpose of small molecule pharmaceuticals, biopharmaceuticals, helping ensuring that the water we drink and the food we eat are safe, and discovering next generation chemicals and materials for use in industrial settings. We leverage technologies such as liquid chromatography, mass spectrometry, multiple angle light scattering, and other advanced characterization techniques, and bring all of those high degree of scientific techniques available to analytical chemists around the world.
Awesome. All right, so now everybody in the audience should have a sense of who our panelists are. So if you have questions, just send them right on over and then we'll get them into the mix. But just to get us rolling, Thras, did you know that you would be working closely with so many partners to bring this project to life? And how did you go about choosing the vendors that you wanted to work with?
Yeah, that's a great question, Hayley. And I have to say that when we were starting this, around 2019, 2020, we had a very naive thinking about, how you build an automated system and how you integrate it. We were, software builders, and we knew that you need someone like, let's say, AWS. But going into hardware and automation and the whole end-to-end integration, we realized that it's all about the partners that you select in this process to be able to go from something that seems impossible to something that you actually may have a plan and approach on how to do so. So, we ended up working, I think, with more than 10 different vendors, including, of course, Biosero and Waters. And I think that it's what makes or breaks a project. In our case, we need to solve a ton of challenges in automated purification. How do you, get the plate and start the fraction collection all the way to how do you program it in such a way that you know which columns to use to collect. And Waters has been instrumental in having these collaborative discussions and trying to figure out these answers. And then, of course, Biosero had helped us from day one to think about the general footprint of the system. What needs to be automated versus what needs to be just integrated in a way that can be scheduled with GPG, and allowed us to think about also how can we take it from a semi-manual, semi-automated process all the way to a full autonomous system. So I would say that it is, it was both a surprise that we needed to work so closely with so many different people, ordering all different parts, getting in the same place. But in the end, also a great process that I think you can build very complex systems with, a lot less complexity than if you would do it yourself.
Awesome. Dustin, I wondered if you could tell us a little bit about the process of designing the solution. Like, how did Biosero contribute?
Yeah, great question. So in the beginning stages, DeepCure approached us, of course. We consulted with them to figure out the scope of the automation project, what we needed to build, what we wanted to accomplish through that. So we need to know the workflow requirements, what devices we want to use to execute that workflow, some of the data handling aspects, what we need to manage there, throughput requirements, storage capacity on the system to meet that throughput requirement, all those things, budgetary constraints, timelines, all that goes into scoping out one of these projects. We then work with DeepCure to design a platform that's going to meet all those goals. And then we'll iterate upon that. We have a version 1, version 2, and refine it over time, of course. But once we mutually agree on the platform we want to build, our project team then takes over from the formal design, the initial build, and testing, method development, all the way through the life cycle of the project to factory acceptance testing and site acceptance testing, where we build it up in the lab, test it out, and hand over the keys, if you will, to DeepCure to get the system into production.
Well, thank you. Andrew, same question for you. How did Waters get involved, and what was your goal in supporting DeepCure?
Yeah, so it really kind of started with a personal connection with a number of folks at DeepCure, and them reaching out looking for assistance in achieving their grand design for this automated synthesis and purification work cell. So we started the conversation with a discussion of what are the end goals, what are you looking to accomplish, so that we could configure the best solution for them, given the vision that they were looking to accomplish. And so from there, we configured an auto purification system for them that's based off of, off-the-shelf components, tied together into a unique installation. And at the end of this story, we brought forward a auto purification system with the 3767 auto sampler and fraction collector, a QDA for mass-directed purification, which really helps with the throughput and ensuring that the right compounds are being purified by the system, while also providing a very rich data stream to the AI, as Thras pointed out, and the ability for the AI to consume massive amounts of data. We brought in a mass spectrometer, a single quadrupole, to add that data fidelity to ensure that they are getting the plenty of data in order to help design new drugs. Brought it all together, and then shipped it off to Biosero in San Diego for kind of initial fitment engineering and testing for implementation, before that work cell was completed and then installed at a DeepCure facility. So it's multiple layers. There's lots of communication throughout this entire process, and kind of played our part as far as helping DeepCure to achieve their vision.
I think that's a good segue into my next question, which is, for anybody on the panel, can you talk about the pros and cons of working across so many teams? So Thras, you mentioned that you'd worked with potentially 10 vendors on this project. What are the pros and cons of that? And do you have any stories that you could share that bring that collaboration to life?
Yeah, I know that's a great, second part of the previous question. I think that some of the pros, as I mentioned before, when you have a project that is very complex in nature, you have multiple moving pieces, you have to go through a pipeline that involves, potentially tens of different steps. You need to be able to delegate the problems into the people that know best and are the experts on the individual steps that you want to work at. It would be, I think, some would say, why don't you build a single device that's integrated and does everything in between? But the reality is that anywhere from reformatting a plate to purification to orchestrating how you move from instrument A to instrument B has its own unique challenges, and being able to work with partners that have done it tens of times or hundreds of times before give us the confidence that at least that piece of that item in our end-to-end pipeline would work as expected. I think that on the con side is pretty much the flip side of what I discussed. So having to integrate and have all the vendors you're working with thinking the same way about your general vision and approach that you want to have in this automation is quite challenging. And you start on a kind of an off-the-shelf idea. What can you take exactly as is and use it, and then you have to meet halfway there on what modifications can we make so that we can make it work in the way that we envision the automation of that system. For example, how do you open the instrumentation to receive well plates for instruments that don't have that capability to be automated? Or how do you use the APIs that may or may not exist? Because they don't exist, how do you build the APIs to get the right amount of data and the right level of details of data so you can drive your AI system? So that is a challenge. And the cons were, having to go through multiple, steps of conversation until we can get where we want.
Andrew, Dustin, any thoughts? Pros and cons of working across so many teams?
I think there's, involving, lots of different companies all working together to help, achieve a vision for a singular client, in this case, DeepCure, takes, fantastic degrees of communication and an open communication amongst all those companies. Because, as soon as something is not appropriately communicated, the house tends to have a, faulty foundation at that point. So I think that's one of the challenges. The good news is it's very easily solved, with the, integration of our day-to-day life with tools like Zoom, as we are using today, or Microsoft Teams or other resources. It's very easy to communicate, whether that is a physical space limitation for the equipment and the resources that it needs, whether that's an air supply or an electrical drop, whatever that is. It's easy to communicate and put that information out there that enables everyone to come together in order to achieve that vision. Whether it's software integration, as Thras pointed out, APIs is something additional needed in the software. There's, a plethora of situations, and all of it starts with, effective communication. And if you have that foundation of effective communication, inevitably there will be something that needs to be addressed. And if you have that solid foundation, you can see it addressed. Without it, it's going to be near on impossible.
So, yeah, go ahead, Dustin. No,
Yeah, just to
go for
expand
it.
on that a bit, sorry. You touched upon project scoping, and that's one of the key things for us from the automation side. The better we can scope out the project, early on, and get as many of the details early in the process, it just makes for a much smoother project delivery, downstream. So one of the great things about lab automation, some of these custom platforms, is it's never been built before. So it's very exciting, but can also be a challenge. You definitely learn things along the way. But to Andy's point, the open communication is definitely a huge plus, and I think we're lucky to work with such great partners and vendors like Waters and DeepCure here. So it was definitely a positive experience.
Awesome, thank you. So I'm really excited to say that Brian Engelhardt, he broke the ice. He asked the first question from the audience. So I want to throw Brian's question into the mix. So different physical sites, for example, US versus Europe, they often have slightly different processes and equipment, even within the same company. How does DeepCure AI deal with different physical systems that need to result in a standardized solution? Thras,
Yeah,
I feel like it's for you.
That's a great question. And I think that, what was the qualifier word, slightly? I would remove that slightly. There are cases where there is a fundamental difference in the way that you have to either orchestrate the experiment or, think about the individual parameters. For example, we have now experience of working with both Tican and Hamilton, liquid handlers, and each of them has their own processes that can achieve the same result. So from the perspective of DeepCure, what we've done very early on is write a data layer where we could build our own models for what are the data that we expect from the processes, and also kind of write a procedural code in our internal software suite, in what we call Galactus, that abstracts the instruments in some abstraction layer that we defined. So for example, the definition of a plate versus a container versus whatever else you would use to transfer liquids essentially is liquids with preference with some IDs. So having that abstraction layer act as a ground truth of what do we want to schedule, and then what do we expect on the output, allowed us essentially to be able to cater the specifics of each individual API that was introduced in the system, and be able to make some sense on scheduling the work list. And I have to say here also, I give credit, to Biosero and GBG. I think they have, it's their prototypical example on why you need a scheduler like GBG to be able to standardize and integrate with all the different pieces of equipment in a way that allows then us to care about a single work list at the end.
Thank you, Thras. So let's take one of the questions from the list that people submitted when they were registering, just to mix it up as well. So the first question there is there an example of a drug that has made it all the way to the market that was identified using only in silico tools to originally identify it?
I can take that. I mean,
Yeah,
talk about
go
that
for it.
can talk about that for hours, but I think, I will try to narrow down the question on what probably is the real question. In silico tools in general have been around for 50 years. And whether it's computational chemistry tooling or bioinformatics, I would say that every drug in the past 20 years have been identified with the help of in silico tools. Now, what I think the question here is maybe introducing AI in the mix. Was there any AI designed drug or a drug that purely the in silico nature of grinding data triggered its discovery? And I would say that I don't think there is an AI drug, if you would call, in the market at the moment. You have companies like Recursion and Silico with multiple drugs in the clinic. If you look at the press, they would say one of these two are the ones to have the first one. We have multiple drugs on the way to the clinic that were designed by AI together with medicinal chemists. But I think it's something that it's also a question that I don't think we care at the end so much. I think what we care about is to see or to be able to quantify what's the value add that you get from the in silico tools in the process. It doesn't need to be just in silico tools. There is no oracle that you give data and the drug comes out. So being able to have a co-creative process where AI and scientists work together and discover the next generation of drugs, that's what we focus on.
I'd completely agree, Thras, and I think you said it best. It's the co-creative process. We turn the time back, at 80 years, and it was purely, human collaboration. Starting in the 80s and 90s, we started adding the computer, and we're just seeing that gradient shift to more in silico support and co-creative process for the medicinal chemist, for the drug discovery scientist. As things move forward. And I think, we're not at the end. The gradient shifts day by day. And what will that eventually look like? I can't predict the future. None of us can. Otherwise, one of us will have won the Powerball by now. So,
awesome. Okay, I'm going to turn it over to Sarah. Sarah has a relatively complex 3-part question, so I'm going to ask it in pieces as we go. So Sarah says, thanks for the event. I was wondering if any of the panel's associated companies can answer the questions I have regarding novel discoveries, such as question 1, do you offer in vivo or in vitro toxicity analysis services, acute, subchronic, or chronic under GLP conditions?
Allow me to briefly try to answer all three. I think that meaning on our end, we're a therapeutics company, so we don't offer services. We discover the drugs, and I think both Waters and Biosero may have worked with a lot of partners that CROs essentially that offer these services. I'm not sure if Andy or Dustin.
Waters, we're not a contract research organization. We do not develop or help develop new therapeutics. We provide the instrumentation, consumables, and informatics used by companies like contract research or like innovator pharmaceutical companies like DeepCure might leverage, but we do not offer services to that end.
And same here. Biosero would be happy to build automation platforms using our Green Button Go scheduling software to automate some of those processes, but that's not something we would do ourselves.
Thanks for your question, Sarah. So Thras, maybe we'll start with you. What is your advice for anybody who wants to follow down a similar path here, working across many businesses to bring a solution to life?
Yeah, great question. I would, let me start by focusing on the automation piece first, or systems like the one I just talked about. I think one of the questions that we were, because the best advice we got is asking yourself whether you need that extra layer of complexity at every given step. So this may be, I have a manual process, it works. Do I need to automate it? If I need to automate it, do I need to have a robotic arm that integrates everything together, or is it okay for me to take the play from one step to another? If I have, a single work cell versus 2 work cells, do I need a robot to take the place from one to another? Within my process itself, do I need to think about all 10 possible, cases of chemicals, or can I actually focus on one first and then go to the next? And the answer to that is that always do the simplest, because that's how you maximize the amount of information you learn. You can debug things. We say during the process, you can figure out which partners you're actually happy working with, instead of trying to do everything at the same time with a project scope that looks like it's 20 pages. So my first advice is simple project scopes. Start bottom up, start building your infrastructure, whether it's software or hardware, and let the users and the scientists interact with it and kind of guide its next steps. And then this way you can actually increase the value and quantify it as you go that your system adds, and makes, to the point I think both Dustin and Andy made, makes the discussions with the vendors that you have to work with so much easier, because you know what you're looking for. You can get the best advice from them versus shifting the scope and then trying to figure out what you're doing.
So Andy, Dustin, do you have similar thoughts along these lines? Like, what advice would you give a scientist who's looking to do this type of automation, or what advice would you even give another vendor on how to look at collaborating across these businesses?
I think it's really important to bring, all the stakeholders to the same table. Once an organization with that vision for an automated work cell, it has selected its preferred vendors, making sure that those vendors are speaking to each other is absolutely crucial. Bringing the scientists from, the company to the table to talk, very serious what science is looking to be done. And, what are the needs, not only today, but 5-10 years for the automated work cell is going to be very important, because, we don't want a decision made today to have to be reevaluated 6 months or a year from now. So understanding what the future might hold for the automation and the work cell is going to be really crucial. And again, open communication. I can't say it enough. With DeepCure was crucial for success. I know there have been other conversations, with Biosero and Waters at the same table for other organizations, and making sure you have everyone there. I mean, we've been in plenty of meetings where there's 30, 40 different representatives from different companies all on the same call, or all sitting around a very, very large conference table in a hotel. But that's what it takes in order to be successful. You don't want to start having these open conversations 8 months after you kick off a project and after you've already made some decisions. That's when you hear the oops, or sorry, that's not possible. And those are some very expensive words to hear 8 months or 12 months into a program.
Yeah, and from the Biosero side, I totally agree with everything Andy and Thrasyvoulos have said so far. Just expanding on that a bit from our side, the automation side, looking at the workflow, the more we can do upfront early on to nail that down, we can then work with you to decide how we're going to manage that, what instruments we want. If maybe we're unsure what the best device is for this particular situation, we can help you get something that's easily automatable and that we know we have a good experience with. Sorry, Teams is going off in the background. We can also work with you to nail down the goals of the automation project as well. So what are the goals of what you're trying to do? Is it just maximizing the throughput, getting more sample, more data? Is it maybe just lights out automation, just more walkaway time for your scientists? So, I mean, those are definitely great use cases for automation. So we can definitely help walk you through that and nail down what exactly we want to achieve with the platform.
Awesome. So I'm happy to say Sarah and Brian have added some more questions to the chat. So let's hit it. The first one from Sarah is, may I ask if the panel is aware of whether AI is currently being used to address these types of preclinical development questions, such as toxicity prediction, PK/PD modeling, or IND enabling assessments?
Yes, it is. Well, short answer, yes, to various degrees and at a number of companies that I'm aware of. Yes.
Sarah says awesome. Okay, from Brian, multi-instrument Microsoft Windows-based systems suffer from unexpected programming conflicts. There is a need for PLC, program logic controller like stability used in the manufacturing space. Is anyone working on an AI solution to make the automation systems more reliable?
That's a great question, Brian. I think for the Biosero side, AI is definitely part of our vision going forward. I don't know if that's specifically around PLCs. I'm not sure if that's in the pipeline at the moment, but it's already, AI in general is something we're using definitely on the applications team already to help shorten timelines, get to the finished product sooner. So I'm sure it's coming, I would say.
Thanks, Dustin. Okay, so I'm going to pull from one of the questions from one of our registrants. And so this question is, how do you think about evaluating whether you need a full automation solution without pulling the trigger and doing it? Chemistry has a long history of being done without a lot of automation.
Yeah, that's a great question, and I think I touched briefly upon it. I think that the way that I would approach it now, with a lot of hindsight, of the past few years, is that one decision is, do you need the process to be faster or higher throughput, or both? Like, what are you looking to scale up? If you're making the exact same reaction and you want to make it 1,000 times, which is something like you would do, for example, in when you have an approved drug and you want to scale it up, that's a very different problem fundamentally than if you want to make a single reaction or 1,000,000 reactions one time. Both would result in 1,000,000 products. The one is exactly the same compound in every well. The other is 1,000,000 different products in each well. And these, if you would put those two extremes on an axis with every other in between, they have different ways that you can automate each step. In the super automated way, you can go thinking directly about an integrated process, a continuous flow process, or whatnot. In the other side, you have to start thinking about it step by step. I have to do purification if I want to do multi-step. How can I automate purification? Can I integrate it with a robotic arm, or is it something that a human should go there? What's the parameterization I can do? How many instruments I need? These types of questions, focusing on each step, can start building a frame of reference on the level of automation we're talking about. And going back to what I said before, simplicity here is the rule. Start simple, identify the bottleneck in the current iteration of your process, and then try to solve that bottleneck with an extra level of automation. So that would be my first advice there.
Awesome. Thanks, Ross. So maybe similar to that, is there something that you would do differently if you had the chance to do this again?
Something I would do differently. I would say something that actually goes beyond the scope of the chemistry, is that we try to automate at the same time both synthesis and screening. And that resulted in very diffuse attention to solving the fundamental challenges of each station, which actually after we focused specifically around synthesis station, that's when we unlocked, great results like the ones I saw with Paxlovid and made it much easier it's much easier to solve one challenge at a time. So that's one thing that I would do differently. And then, yeah, I don't think anything else. I think that the communication aspect, as Andy said, was the fundamental piece that allowed us to do not have regrets. Because if you're upfront and you're like, this is what I'm expecting, can I actually get it? This is the way that you don't regret or do things differently afterwards.
Awesome. So I think we have covered all of the questions that have been submitted to us, and we've covered quite a few beyond that. I would say that brings us to the close of our time. I really want to thank so much all of our panelists, Ross, Dustin, and Andrew, who are sharing their time today and their thoughts. And I want to thank everybody who has joined today and asked questions. And with that, I'd love to turn this back over to Gina to wrap us up.
Great. Thank you, Hayley. I just want to echo your sentiments. Thank you all again to the panelists for participating today. very grateful for your insights and your knowledge. This recording will be shared on Applied, which is SLAS's e-learning website, and will also be emailed to you all directly after the conclusion of this event.
Thanks, everybody. See you
Thank
later.
you all so much.
Very much.
Everyone.
Thank you, everyone.